distinguished speakers. The first is Dr. Petra Fromme, a director professor Biodesign Center for Applied Structure Discovery and School of Molecular Sciences at the ASU. Petra received her bachelor's degree and a master's degree in biochemistry from the Free University of Berlin and a PhD in chemistry at the Technical University of Berlin. She was assistant and associate professor at the Max Warmer Institute before joining ASU as a full professor in the School of Molecular Sciences. She was appointed by the ASU President Michael Crow as the Director of Center for Applied Structure Discovery in 2014. Her research interested in the study of structure to function relationship membrane proteins involved in bioenergy conversion and infectious diseases. She is an integral part of the team of SU researchers and the international colleagues that developed the technique of serial femtosecond nanocrystallography for analyzing proteins using high-intensity X-ray free electron laser. Now, she has published over 200 articles and is a nationally recognized leader in the photosynthesis, protein macromolecule crystallography using synchrotrons, and also protein nanocrystallography using XFELs. Okay, please welcome Dr. Fromme. Please take away, Petra.
Yes. Thank you very much for this really kind introduction. So can you see my screen?
Yes, yeah.
Yes, okay. So I want to bring you into a different type of research, which we are doing. So we are not directly involved in clinical research, but we are developing new techniques which could make a revolution in structure determination and drug development of important medical targets. So I will tell you today about femtosecond crystallography towards molecular movies of biomolecules in action, and what this would mean for their potential for novel drug discovery. So when you look at what we are doing in our center, we aim to develop new revolutionary techniques. That means techniques which are unique in the world, here in Arizona, to get structure and dynamics of biomolecules, making new discoveries, both in medicine and in energy conversion. And this is something very new, because when you look about structures, 99.99% of all structures determined from biomolecules, including medical targets, are static pictures. But when you really want to develop a drug, you have to know the transition state, you have to know the mechanism. And that means you have the dynamics of the molecules. But all structure determination methods are actually static. Both cryo-EM and X-ray, standard X-ray crystallography require freezing of the cell to mitigate radiation damage. Thereby, with these methods, you cannot get the dynamics, get a static picture. And so what we want to do is to get molecular movies of molecules in action. And I'll tell you today what we are doing, trying to do to reach that goal. So the center has a large number of faculties. So we have 17 faculty here. And we have people working on these new technology developments, which you see on the right side, including my longtime collaborator, John Spence, who unfortunately passed away this year. And then on the left side, you see the application sites. We have everything from cancer to biology, immunology, G-protein coupled receptors, which are 40% of our targets, and also photosynthesis. And even if we are now talking about the biomedical relevance, I want to just start with actually the motivation coming from photosynthesis. Because this is a picture, simplified picture of photosystem 1, which is still the largest and most complex membrane protein ever crystallized. And it took us 13 years from the very first small crystals to get a near atomic structure from really big ones in 2.5 angstrom with standard crystallography. And just an idea, the photosystem consists of 36 proteins and 381 factors. And this is just a simplified picture. And this is a big sun catcher capturing the light from the sun and converting it in energy. So making the plants green. And this is one of our to unravel the mechanism of water splitting and photosynthesis. But we need new techniques to determine not only an atomic structure, but also a movie of this molecule in action. Ideally, not taking 30 years or really big crystals, but takes the very first small crystals, which are so small that you even see them under the microscope for structure detection. And for this, we need a new tool, and this is an X-ray free electron laser. So I can tell you a little bit what an X-ray free electron laser is. This is a picture in the accelerator hall of LCLS, which was the first free electron laser in the world, built in Stanford and started operating in 2009. And these X-ray free electron lasers are incredible machines. They make X-rays which are so strong that they destroy any solid material. So when you just compare Dark Vader and his lasers are completely peanuts against that free electron laser. They not only destroy the molecules, they actually destroy the atoms. So they rip off in about 10 femtoseconds the inner electrons. So then the rest of the atom is positively charged, and then it explodes in this kind of Coulomb explosion because all the atoms repel each other. So what do you think is, now X-rays are damaging, so this is even more damaging. So what is the benefit of an X-ray free electron laser? The X-ray free electron laser makes so incredible short pulses that you get a picture of the molecule before it is destroyed. And this was actually a simulation from Neutzer and Heide from 2000, which proposed that when you would be faster than 10 femtoseconds, you can get a picture of the molecule before it is destroyed. And thereby you don't have to worry about radiation damage anymore. You outrun the radiation damage, and you can get pictures of the molecule in action at room temperature. And to give you just an idea how short a femtosecond is, a femtosecond is 10 to the minus 15 seconds. And just so the idea is the time difference between a femtosecond and a second is the same as between a second and 32 million years. So we have proposed actually already in 2003 to make really, really small crystals and then shoot them through the X-ray beam at room temperature and then get a structure from these molecules on the fly. And then we can excite them, we can mix them, so we can get a molecular movie of these crystals practically flying through that X-ray beam. But we get no funding in the first 7 years because the referees doubted that you would get a picture before the molecule is destroyed. They also doubted that we could evaluate the data because we get only one snapshot from each crystal before it is destroyed. And they also doubt that from all these 10,000 or 100,000 of individual snapshots, you would get a structure. And then when we did the very first experiment in 2009, just 2 months after the accelerator opened, we used actually not a small model system, we used Photosystem 1 as a model system. And what you see here on the left side is you jet the crystals through the X-ray beam, and you see this glow here. This comes actually from the plasma form when the jet hits the beam. So the temperature at the end is higher than the inside of the sun, but you get these beautiful diffraction patterns and you get a really nice structure. And the first one was at 8 angstrom because the energy was not high enough when the accelerator opened to get atomic resolution structures. But in the meantime, we published more than 30 papers in Nature, Science, Cell for molecules in action using this X-ray free electron lasers. I want to show you just now a medical application of it. And this was actually a very interesting story because it is a protein which is involved in degrading your red blood cells when you get infected with sleeping sickness. So this is called CAT, and it was crystallized in a very odd way, interesting way. It was crystallized in living insect cells. So often when you want to make a protein for structural determination, you have to express it. And eukaryotic expression hosts like insect cells are much more similar to, for example, human cells than when you would do this in E. coli. So when our colleague, Lars Redeke, expressed this protein in cells, saw something in an electron microscope which looked like a crystal in the living cell. Isolated the crystals and shoot them through the beam, and we get the very first structure of protease with the pre-peptide bound. And the resolution is so good that we can even see the glycosylation really nicely. And now there are drugs under development which mimics this peptide, which inhibits the protease when it is still in the mosquito. And then get cleaved off when it reaches your red blood cells. So that might be a very promising target against sleeping sickness. And this was highlighted as one of the breakthroughs of the years in science. It was not the breakthrough because that was the Higgs boson. Nobody can beat the Higgs boson, but we were one of the 9 front runners. So we have followed up on this research in the meantime because we wanted to see the mechanism of the crystal growth. And now we label protein actually in the development of Drosophila with a fluorescent tag. And then you can actually see the crystals in the living insect cells really nicely. And we can embed them in this media and directly deliver them to the X-ray beam. Means we don't even have to crystallize the protein. We can express it in the living insect cells, and we can deliver it to the beam and directly essentially get structures from these crystals grown in living cells. This has not only an interesting aspect for the structure determination, it's also very exciting because we're wondering about the mechanism. The first 1 or 2 days when we express it, the complete cytosol grows. But as soon as these crystals form, the cytosol is completely dark, but you still see that the crystals grow in the living cells. So how could be this mechanism when the protein is not in the cytosol? How can the crystals still grow? And so when we look at electron microscopic pictures of these crystals, we see that actually ribosomes, polysomes are attached to the crystal. And you see the really big ones still have polysomes bound. The small ones which stop growing have actually polysomes close by, but have detached from the crystal. So we are wondering, we have published this just last year in ACDD. So we are wondering if maybe aggregate formation, not crystal, but aggregate formation in neurodegenerative diseases like Alzheimer may have a similar mechanism, that as soon as a prion protein as aggregates form, the ribosomes may dock to the surface of the growing aggregate. And thereby the protein is never really folded in solution. And so we got just now seed funding for a new research project with Paul Kuhlmann and Carol Husby to study the mechanism of tau pathology, actually both in insect cells, human cells overexpressing tau, and also in patient brains biopsy samples to investigate if this mechanism may play a role. Because if so, one could probably see if one can inhibit the binding of the ribosomes to the growing aggregate in Alzheimer's disease, and thereby see if one can block the formation of these aggregates. Then we did more time result studies where we really now gone from structure determination to molecular movies. And I just want to show you 2 examples of our recent work here. One is a paper which we published in Nature in 2007 of dancing of a riboswitch. So riboswitch regulate gene expression, and they're also involved in several diseases. And they switch between distinct RNA structures in response to binding small molecule metabolites. And so when we mixed then the crystals with adenosine, just before we deliver them to the X-ray beam, then we collect data at different time points. And you see that there are huge conformational changes in the RNA riboswitch upon this mixing with the effector molecule. And it's not only affecting the RNA, actually the molecules moves by 30 degrees within the crystal. And when you try to do this with big crystals, the crystals just crack and dissolve. Whereas a small crystal can accommodate these changes. And when we looked at this with AFM, so Dr. Wang looked at it with AFM, he sees actually the molecule fusing in from one side, and then like a domino, all the molecules in the crystal change their conformation layer by layer. So this also shows that you do time result studies with even large conformational changes in crystals, which was before deemed to be impossible. When we look now at enzymatic reactions, this is an example of looking for fighting antibiotic resistance. This is one of the really nasty proteases which degrade antibiotics. And we have essentially developed a mix and inject device. So we have the crystals here in the center, you mix it with the substrate, and then you can get with millisecond resolution the real mechanism on the enzyme. And what you see here is that the substrate binds first at conformation A with a relatively large distance to the serine molecule. Then there's a rearrangement, and then you form actually a covalent bond at about 100 milliseconds in the catalytic process, and then later at 2 seconds and 5 seconds, cleaving this bond. So you can really see all the details of the conformational changes in the catalytic site. And then you can develop, hopefully, new antibiotics, which essentially mimics this transition state and thereby might be essentially new drug against fighting antibiotic resistance. So that's one example where we really use mix and inject for these technology developments. We heard in this really interesting talk before the break about cancer. And our center now became involved with the Center for Membrane Protein Drug Discovery into the Chemical Biology Consortium of the National Cancer Institute. So that's a major consortium from the National Cancer Institute to develop new drugs against cancer. And we are one of the 20 centers here in Arizona. And the first project which we got involved is actually a protein called TASPase. It's a threonine protease, which drives cancer cell proliferation, invasion, and metastasis. And there was a structure of this protein, but it was a truncated version. And this truncated version was inactive. And so they really used drug design based on this inactive mutant, which could bind drugs, but doesn't cleave in native, to develop new drugs based on this. But this doesn't went to anywhere for more than a year, because obviously the native structure, the active structure must be different. So having the structure with an inactive enzyme doesn't really help you. So because it was a tricky subject, we wanted to get the structure of the full-length, fully active TASPase. And this is actually one of the challenges of the XFEL research, is that there are currently only 5 XFELs in the world. There's one more in Shanghai, but it's not opening yet, very likely next year. And so in Stanford, 2 in Germany, 1 in Germany, 1 in Switzerland, 1 in Korea, and 1 in Sakhalin. And then in 2019, LCLS found for 1.5 years to be able to build a second accelerator parallel to the first one. And so we had to travel all over the world to collect data on the TASPase. And so we went to Korea, South Korea, XFEL is actually here the team collecting data. And we got really nice crystals with very nice size distribution. And we got the first of the full-length enzyme. And here was actually the first structure of the intact enzyme. And the really interesting thing is that it doesn't form just a single heterodimer. It forms a huge hexamer looking like a double donor. And the parts are cleaved off to crystallize it before it actually connects these 2 parts of the double donor. We got a really nice structure with the parallel XFEL. In the meantime, we got also nice synchrotron structure, which we have just published, and we are going to publish this soon. So what does it mean for the drug development? We have the first view of the site, active site of the full-length protein. And when you look in detail, then you see here the structure of the active enzyme in green and the one for inactive enzyme overlaid in orange, in pink. And you see huge conformational changes actually upon the activation. So for example, the binding site is actually closed in the inactive state, whereas it's open, this is moving out here, in the active state. And so what we are currently trying to do, and especially like Stanford has now opened again, is determine the structure with drugs and also do this mix and inject experiments, which I showed you for the antibiotic resistant protein, to get time result studies and to get the catalytic mechanism of this important drug target in the future. So then you probably know that G protein coupled receptors are very important targets. So 40% of all current drugs targeted to them. But they are membrane proteins and thereby notoriously difficult to crystallize. And then members of my center, especially Valio, developed together with Vadim Sheretsov at USC, crystallize actually the crystals in a lipid phase. That means in a membrane environment. But this thing has a consistency of toothpaste. And when we first proposed time of the experiment on these G protein coupled receptors at an XFEL, we didn't get time for 2 rounds of applications because they told us you will never get that toothpaste to fly. But we developed a special injector which extrudes these crystals and also needs much less samples than our standard liquid jet. And we get multiple structures here. This is, for example, a picture of the angiotensin receptor, and this is also this drugs bound. We had, I think, 3 or 4 different drugs bound also in clinical trials to this receptor. And then we have, this is smoothen receptor, which is also very interesting for cancer, bound with cyclopamine. Cyclopamine is actually a drug which was discovered because cows feeding on corn lilies got lambs with only one eye and severe neurological defects. So they died like less than a few hours after birth. And because of this one eye appearance of the little lambs, of the little cows, it was called cyclopamine because they look like cyclops. So cyclopamine itself is dangerous, but variations of cyclopamine have now in clinical trials for cancer fight, because it is actually inactivating the smoothen pathway. So this might be something really interesting. And they tried unsuccessfully for multiple years to get structures of this cyclopamine, and they didn't, because the cyclopamine changed completely this extrinsic part of the receptor and makes it extremely flexible. So you cannot grow any big crystals, but we can call this really small crystals for femtosecond crystallography. And we are at the moment also labeling drugs with fluorescent labels, which can be cleaved by light. So we can make the time result studies with a light-driven system where we would release a drug upon a UV flash and then look at the conformational changes on the GPCRs. And then when you get a dynamic structure of the transition state, then you can screen the drug libraries with a virtual screening, which then you identify the most promising future drugs, and then you combine this with binding assays. And so we have shown that this kind of virtual screening combined with the dynamic structure of the transition state, you can actually get in less than half a year the binding affinity from micromolar to nanomolar of your new drug. And then when you have less drug that you can have to apply, then it's also less chance that it makes toxic side effect in the liver. So some of these developments are now in preclinical trials. So at the end, from the scientific part, I want to actually show you our recent research on a protein involved in the COVID virus, so SARS-CoV-2, hiding from the immune system. And for my talk, it's now important that SARS-CoV-2, that means the cause of COVID, is an RNA virus, and all the RNA viruses come practically with the messenger RNA ready to use. So it has a cap, the 5' end, and a poly A tail at the C' end to be able to recognize by the ribosome. And so when the RNA is uncoated, when the virus has infected the cells, then the proteins which have enzymatic functions, so for example, the main protease, the enzymes involved in RNA replication, and also our protein nanolure, are made in the first round from this first transcript. And then a negative strand is formed, and the negative strand is then formed as a blueprint for making the messenger RNAs of all the structural protein, including the spike protein, and also for replicating the RNA so that you make a positive strand which can be packed in the virus. So when now the positive strand is converted into the negative strand, then the poly A tail converts into poly U header. So this negative viral RNA strand contains a poly U header. Poly U headers don't exist in the human cell. And so their signature for to detect that the cell infected with an RNA virus. So as soon as the immune system detects this poly U headers, there's a big virus alert, and the complete immune system, macrophages, and everything is activated to fight the virus. And we all know that COVID is now a pandemic compared to MERS and SARS. The system is late in detecting it. So this is the reason why people spread the virus, and still now even at a very high rate, before even becoming sick and showing any symptoms in case of, for example, fever. So the delayed immune response is at the core of why essentially COVID is a pandemic. So what is now the function of nanolure? First, people thought it would be involved in replication, but then just discovered last year that it actually cleaves off this poly-U header and thereby hides the virus for a while from the immune system. So we really want to get the mechanism of this protein, because if we know it, we could hinder NanoU to work, thereby people would get infected, would immediately fight the virus, thereby the infection would be not as serious, and one could easily detect people who have been infected. So we have actually made crystals. We overexpressed it, and we made crystals of the protein and tried different substrates. And the protein in the crystal is fully active in the neutral pH range in cleaving actually the RNA. And so we went then in the time of COVID. This was just the second experiment taking place after the free electron laser in Stanford Open again. So we drove all the way, my student and I, to California, to Stanford to conduct the experiments there in COVID. And they had very limited access. So only 2 members of our team could attend. Normally we attend 30 people from our group, one of these experiments. So this was a very limited team. We were very successful, and we got the first structure, room temperature structure of essentially NAN. And we had it actually with citrate bound in the binding site, which was pretty interesting. You know, citrate is in a lot of citrus fruits, so maybe citrus may help you essentially in the fight of COVID. But we want to know, essentially in the next studies, want to see how the RNA binds. And so we established new crystallization conditions without citrate. And we also found crystal packing, which has huge cavities, because we have to bring the substrate into the crystals. So this is more than 100 angstrom large shells here, the 2 catalytic sites. And so this is ideal for future SFX studies. And we have beam time coming up at the free electron laser end of the year to do these time of Earth studies. So the determination of the catalytic mechanism will be a big deal, because it could form a structural basis for development of new drugs inhibiting NanoU. And it is very specific for the pathogenic versions of coronaviruses. So maybe it's a key of why essentially COVID is a pandemic. So one of the disadvantages of essentially all this free electron laser work is the access, because I told you that there are only 5 free electron lasers in the world, and each of them can only do 1 experiment at a time, because it's a linear accelerator. So each of these big accelerators, the 5 in the world, are 1 kilometer long. Actually, the one in Hamburg, which is the newest one, is actually 4 kilometers long. So it actually starts in Hamburg and ends in a different state of Germany. And so what we are doing now is a pretty crazy thing. We try to shrink the X-cell from 1 kilometer to 10 meters and build the first prototype for, instead of 1 billion, for 20 million here at ASU. And for this, we lured Bill Graves away from MIT, worked more than 20 years on the development of compact X-cells, but he never got the money to build one. And we combined it with the exciting bioapplication and are now bringing the first compact X-ray sources to short pulse to US and ASU. And we do this in 2 stages. First, we built the compact X-ray light source, which has about 300 femtosecond duration. And then in the second stage, where we just submitted a large-scale proposal to NSF, we will build the lasing version of it and will increase the access from bioresearchers to X-rays, custom designed for bioapplication. And the instrument will allow us also to combine it with spectroscopy. And the second version will have sub-femtosecond pulse duration, so we overcome any damage, even electronic damage. So how far are we? We actually, in 2014, when I started with the center, I gave President Crow just a half-an-hour presentation. And I showed him pictures which Bill Graves has sent me from a presentation he made to the King of Qatar, because they really were desperately wanting to build that X-ray, but they didn't get the funding. And so the King of Qatar, unfortunately, didn't want to build one. So I convinced President Crow just in half an hour that this is the coolest thing in the world and we really need that at ASU. And he made me the offer to getting the center and said, we will build a compact X-ray at ASU. So while we are finding a new building, because it's clear that this is a hard X-ray accelerator, you have to have shielding and everything. we will get a new building, Biodesign C, and in the basement, there should be space for the accelerator. In the process, we convince President Co that when you make this multiple $1 million investment, then we should build space for two. Because I told you that LCLS in Stanford was 1.5 years out of working because they built a second one. But when you build the first instrument of its kind in the world, it will never be perfect. You have to optimize it. But when you do upgrades, then you cannot do experiments. So we convinced him to build labs in the basement for 2 accelerators, which here would be 10 meters, so it would be the accelerator, here would be the experiments. Here is a high-powered laser, which drives the accelerator, because the whole thing about it is we replace these magnets. And all the big accelerators, the principle works like that. You have an electron forced on a wiggling path by essentially these 1.5 kilometer long ondulators, which have alternating magnets. Force electrons on a wiggling path, and then the electrons interact with their own X-rays, which they emit. And then the faster one gets slower, the slower ones get faster, and they start to wiggle in phase. And only when they all wiggle in phase, then you make these ultra short X-rays. This is called self-seeding, and all the big X-rays work of that. But in principle, this is technology from the 80s. What we are doing is now technology from the 2020s, because what we do, we replace these kilometer-long accelerators by essentially interacting electrons flying with the speed of light, hitting them heads on with an ultra-strong laser beam in the infrared. And this is about 80 trillion times stronger than your laser pointer. And this collision shoots the electrons out of their path, and thereby it's a short excitation. So in principle, we replace these 1.5 kilometer long magnetic ondulators by an which is the 10th of a human hair. And so we are going to build 2 of these. And I just tell you biodesign building that have this in the basement, and we have on the 3rd floor all the laboratories to make the crystals. And this was when we started the construction in the fall 2019. This is now in the winter. And we have already, this is our now compact light source, really compact, 10 meters long only. And we had the first electrons already in the spring this year, and we'll have the first X-rays when our ultra strong fully commissioned by end of this year, in the spring next year. So stay tuned for that. I want to tell you for all the essentially funding we got. And one person which is not on here is actually Leo Buse, because this first accelerator, which we hopefully have operating spring of next year, is fully funded by ASU. They gave us $7 million and a $10 million donation of Leo and Annette Buse, which actually helped us building the first version, because we didn't also get any public funding first. Because they all said, you have to show first the proof of principle, but how you can do that when this instrument nowhere exists in the world. So the first one really built by ASU and a private donation, and we have now applied for a grant for NSF to get hopefully the second one. And I want to also specifically thank BioX, National Cancer Institute, and also Mayo Clinic Structural Biology Alliance. And I want to mention all these collaborators which we have. There are more than 50 authors of most of our papers, and I know everybody in person because we do the experiments together. I want to specifically mention here Henry Shepman at DAISY, and then also the team at SLAC at Purdue, and here Paul Coleman, which we work now on Alzheimer's disease on these projects. And this is my group at Arizona, and I thank you for your attention.
Okay, great talk and wonderful. Technology is amazing. So we don't have question from audience. So that I have question because I know that this technology requires special, the instruments, how to compare with the cryo-EM, because I'm not this field expert this field. So compare with the cryo-EM, I know there also is another powerful, instruments.
Yeah, electrons are actually 100 to 1,000 times more damaging to biomolecules than X-rays. So they interact stronger and they're more damaging. And so thereby you have to freeze for cryo-EM. And the revolution in cryo-EM to get now atomic resolution structures came from the fact that you have ultra-fast cameras which make a movie of your image. And then you extrapolate to the time before you made the damage. Okay, so this is why now cryo-EM can get atomic resolution structures. And when you just want to have a static picture of your molecule or your complex, then cryo-EM is a method to go. We actually also have faculty in my center working on cryo-EM, and I have myself projects on cryo-EM. But when you want to get a molecular movie, then you have to essentially have a technique which outruns the damage. So that is the major thing. So time result studies are the major application of essentially these X-ray studies. And they're basically complementary actually to cryo-EM. So you can get a nice structure with cryo-EM. You can try to get the molecular moves with femtosecond pulses which outrun the damage and do everything at room temperature.
Okay. Yeah, Bill has a question.
Hi, Petra. Nice to hear the latest. Lots of good stuff as per usual. On the time resolved end of things, I had a question going back to your time resolved protease question. That's sort of a specific one and a more general question. So the specific part is, so it looked like the slow step in that mechanism that you looked at was actually breaking of the peptide bond, or of the compound bond, and that covalent linkage happened very quickly. The specific question is, how does that compare with what's known about the kinetics in free solution? And the more broader question is, how are the time resolved time constants that you're seeing in crystals comparing overall with solution kinetics when we have them? Yeah.
So maybe I can answer the last question first. So we measured actually with the suspension because the crystals are really small. They are less than a micron. So you have very fast diffusion into the crystals. And we had actually two crystal forms. One crystal form diffracted really well, but the kinetics was slowed down by a factor of 15. The other one had pretty big solvent channels. The resolution was not as good, was only like about 2.8 angstrom, but this was practically the same kinetics as we saw in solution. So this is a reason why for the nano-U, we also went with a crystal form which has a really huge channels, so that we can really compare what happens in the cell in solution with what happens in the real world, in the body. So we want to get the mechanism, and thereby you have to unfortunately take most times not super tightly packed crystals. In the worst case scenario, they would be so tightly packed that you don't get the substrate even in.
Right.
So we always test essentially the function in the crystals before we do the time resolved experiments.
Yeah. So, but there's there's the additional possible complication that the crystal context could affect the kinetic pathway, I guess. First you got to get the compound in there. Yeah, that's clearly true. But then you could dampen key loop movement, for example. And I'm just wondering if the field is seeing any of that, or if it's really corresponding well with the solution work.
So the interesting thing is that for many of our studies which we have done, it looks like the conformational changes induced in the protein are stronger than the crystal contacts. Yeah. I showed you the picture from the riboswitch. You know, the molecule makes a huge change and even changes its position in the crystal. It's even a different unit cell after the reaction has taken place. And the really small crystals can accommodate these changes by practically staying intact and the molecules switching layer by layer. Whereas really big crystals, they always crack and are dead practically after to do this. We have also done for photosystem time result studies on photosystem 1 with ferredoxin. And after you shine light, ferredoxin undocks from PS1. So it says, nice to see you, alligator swims away. And so the crystals dissolve invisibly in less than half a minute. But when we did the time resolved studies, we already see that the diffraction quality diminishes in less than 10 microseconds. So it goes really fast. But then we still get essentially a movie from the start of the process. So the electron transfer, and then essentially we see the beginning of the undocking. We don't see the end of the undocking because then the crystals are not there anymore. So it seemed to be the conformational change is stronger than the crystal contact. The nice thing is because you hit each crystal only once, you can even see this irreversible reactions, even if the crystals will practically disintegrate during this process.
Yeah, because we run out of time. Maybe I understand Nina has a question. Maybe after that we can, you have calculated, maybe talk to Petra. Is that okay?
Yeah, I can.
Yeah, we already over the
No problem.
5 minutes.
I'll email Petra, no problem.
Okay, thank you, Petra, for your beautiful, talk and a wonderful science, and hopefully, we can enhance the interaction between, the SU and U of A, because we have a lot of people doing the, the biomedical science. So that's okay. Thank you again.
I can just invite everybody when we have the compact expert working, so then we can start many new collaborations for using the system. And
we look forward to you. Yeah.
You would only have to come over to Arizona State University to do experiments.
Oh, great. Okay, let's move to the